Okay, thanks. If you could all come on in. There's plenty of seats up front. And Greg, can you grab the door if you don't mind? Thank you. Okay, good evening. My name is Dan O'Connor. I want to thank everyone for attending our research reception here at SITC. Really appreciate everybody coming out. So during this presentation, I may be making some forward-looking statements, so please take a moment to review this slide. Next slide, please. So tonight's agenda is really three topics that we want to hit. First, we want to talk about cervical cancer. Our lead product candidate, AXAL Fililizumab, aka AXAL, in cervical cancer. After that, we will introduce Dr. Brian Slomovitz. Dr. Slomovitz is joining us from the University of Miami. He is the lead investigator, the principal investigator, on a combination study of AXAL plus Durvalumab in metastatic cervical cancer patients. So Brian's going to provide insights on his phase 1 study. That's the subject of the poster and a presentation tomorrow. After Brian, Michael Princiata, PhD Listeria expert, is going to take the podium. Mike is going to speak about our neoepitope program, the landscape with respect to neoepitopes, and our product candidate that we call AXAL-NEO. This is the product, thank you. ADXS-NEO, this is the product candidate that we recently licensed to Amgen in August. Okay, next slide. So let's start with the unmet need in cervical cancer. Really, this slide, and the title of this slide, I think, is a gross understatement of the situation with respect to cervical cancer. Why? About 10 days ago, The Lancet reported an updated data. Sorry, Greg, one more slide. Or Raina. Thanks. So understatement is really kind of the theme of this slide. The Lancet recently reported the incidence rate globally of cervical cancer. And you see that number reported here, 500,000 new cases every year. They project another 700,000 cases by the year 2030. That is an epidemic. And really, just to sum it all up, that's why we're here. This is an unmet medical need. There's been precious little drug development in the space of cervical cancer for the last 30 years. The therapies that are available to patients in this setting are surgery, chemo, radiation, and recently in 2014, the addition of Bev, which does what Bev usually does in solid tumors, which added a couple extra months of life and associated toxicities. So I pause to make this point because I don't think people really appreciate the opportunity with AXAL. There's a tremendous unmet medical need, precious little drug development in this space. And I think part of the reason why that's occurred, and this is really just my point of view, is that the advent of the vaccines that have occurred. Those were tremendous advancements to address a world health problem, but really I think they potentially took the pressure off of finding a treatment for patients with cervical cancer. So we know a couple things about the vaccines that exist today. One, we know that the uptake rate is plateaued at about 30% worldwide. There's reasons for that. Secondly, we know that if you are infected with a strain of the virus, you're not a candidate for the vaccine. We also know that 80% of sexually active women are infected with the virus. Not all of those patients will convert into some type of malignancy, but many, many will. In fact, if you can go to the next slide, Rania. Sorry, one back. Okay. Where is that number? So I'll just, So again, the point here is that this is a high unmet medical need, and there's been extreme little drug development in this space. So we think that translates into a great opportunity, not only for patients and their physicians like Brian and many of the other physicians that exist in the United States, but also as a commercial opportunity for Advaxis and its shareholders. That point is really what I wanted to bring home here. I think when I was at ImClone Systems, I developed and helped the team there to develop Erbitux, and I used to say Erbitux is a pipeline in and of itself. Next slide, please. Why did I say that? Because it was one product candidate addressing multiple different types of solid tumors. AXAL is really the same thing. We've got the opportunity with this product candidate to treat many different types of HPV-associated cancers. Our focus is going to be cervical cancer and anal cancer to start. Why? There's, again, no drug development in this space, very few competitors in this space, and we are committed to bringing it forward. So that's actually a sweet spot, I think, for drug developers. Going to a place where there's a tremendous unmet medical need for physicians who really need treatment and treatment options, and where there's not a lot of competition. Cervical cancer, anal cancer are both spaces that we think provide us that opportunity. Next slide. So that was the worldwide situation. The situation in the United States is equally as dismal, really. So we see 79 million people in the US are currently infected with HPV. About 13,000 of those patients will see cases of invasive cervical cancer annually, and about a third of those will result in death, okay? So again, putting a fine point, that is why Advaxis is developing AXAL in cervical cancer. Next slide, please. So I think you can think about it as a bookends approach. We want to be at the earliest stage of the disease, high risk, locally advanced, and we want to be at the end of the disease after patients have exhausted all of their treatment options. This slide gives you a good indication of what the eligible patient population will be in both the metastatic setting and the study that we're currently conducting, what we call aim to serve, in the adjuvant setting. So roughly 5,000 patients in each of those groups. Metastatic melanoma is basically the same type of situation. So that's a good comparator. That's about 5,000 patients in metastatic melanoma. And you hear a lot about that disease, right? So next slide. You stack that on top of the situation in anal cancer. Again, extreme little drug development in the space. Surgery, chemo, radiation are the only treatments that are available, and you see a pretty significant patient population. So again, bringing this together, from our point of view, and the reason I'm spending time to talk about this is we think that the opportunity with AXAL is a very significant one, and we're going after it. Next slide. So data was produced by the company and provided to the company by the GOG NCI that was released by the company a couple of weeks back. I think there's been a lot of information in the news lately. I want to take time to talk about these data because from my point of view, this is the most important data set that Advaxis has put out to date. And I think it creates a tremendous opportunity for the company for an ability to file in Europe, which is what we plan to do. So many people have seen the schema from this study before and are probably familiar with it. I'll take a moment to describe it briefly. It's a traditional Simon two-stage study in metastatic recurrent cervical cancer conducted by the NCI and the GOG. It was conducted under our IND, but it's their study. They designed the protocol. They designed the endpoints. They defined the endpoint as 12-month overall survival. Why did they do that? Because they know, and physicians like Brian knows, that you can shrink tumors for patients in this population, but the most important thing to do is to extend life. And they thought that the endpoint that they wanted to set for this study would be the most meaningful endpoint for them and their colleagues, which is life extension. So they set the primary endpoint of the study to be 12-month overall survival. And as like other Simon two-stage studies, part 1 efficacy and safety had to be achieved in order to graduate into part 2. So what did we see with respect to part 1? Next slide, please. Many folks are probably very familiar with this slide. This is the KM curve from part 1. Part 1 enrolled 26 patients and showed a 12-month overall survival rate of 38.5%. This is the first time in the GOG's history of conducting studies in this setting, and they've been doing so for a decade and a half and have done multiple studies that they achieved a 12-month overall survival in excess of 30%. When I mentioned bevacizumab before, it's been approved as a frontline. The comparator study that was done by the GOG with Bev showed a 12-month overall survival of slightly less than 30%. Sorry, do you want to say something? You're good? Okay. So I think a pretty significant comparator, right? This is Bev, which is now approved in this setting, did not do, or AXAL outperformed Bev in a sister study conducted by the GOG. This enabled the study to go on to part 2. Let's go to the next slide. These are the data that we recently disclosed in a press release with respect to part 2. two points I want to bring home here. One, these data, we're not fully going to go into these data because we're going to look forward to presenting them in a publication and at a major medical meeting, but we can provide the top line data. That's point one. Point two, we're taking these data and we're going to file for a conditional license in Europe. And we're planning to do that probably around the mid part of next year. The slide will say second half 17. Our goal is to do it by the mid part of 17. Why? Why are we taking that approach? Really, it comes down to, from my point of view, this number right here, 25% 12-month overall survival. Where did that number come from? This protocol had a feature where the GOG, a built-in feature, where the GOG calculated at the end of the study what they would have expected those patients enrolled in the study to do from a 12-month overall survival standpoint. That's that number. So they prospectively described in the protocol a way to determine what they would have expected patients who received no treatment, who looked like the patients enrolled in this study, to do from a 12-month overall survival rate. That's 25%. When you take parts 1 and part 2 is 37.5%. Again, part 1, 38.5%. When you take those two together, you get a denominator of 50 patients, and you get a 12-month overall survival rate of 38%. When you compare that to 25%, it's pretty straightforward math. You see a 52% increase in the 12-month overall survival rate. We think that was an important finding. In fact, again, that's what stimulated us to say, let's file. Why? What's available to these patients around the world? Nothing. They've progressed through every available treatment. There's nothing available to them. We know that we know what Bev did in a comparator study, in a sister study to this study. We've now gone above that, and more importantly, we know what patients would have done had they not been treated per the GOG's protocol. The quote here, I think, is probably says it the best, and this is the principal investigator, Warner Ho, who provided this quote. These data demonstrate a meaningful improvement in 12-month overall survival rate compared to the GOG historical studies. Next slide, please. What about other therapies that are in this setting, right? I said there's precious little drug development in this setting. This is one study that was conducted as part of the Keynote study by Merck with Keytruda. These patients, numbering 24, were PD-L1 expressors. So it wasn't like the GOG study where they took all comers. These patients required a certain level of PD-L1 expression. What was observed from the 12-month overall survival rate in this patient population? You see it right here, a 33% 12-month OS. So from my point of view, I think AXAL stacks up really nicely to what Keytruda is doing in the same patient population or a similar patient population. Next slide, please. With respect to safety, part 1 and part 2 is consistent. We're seeing grade 1 and grade 2 fever and flu-like symptoms. These are resolving relatively easily with either self-resolving or resolving with anti-inflammatories and antiemetics. An occasional grade 3 or grade 4. So again, as I look at the safety profile of the drug, you see infusion-related events. So you see fever and flu-like symptoms resolving within a couple of hours, and that's it. No long-term toxicities. I think, again, another strong attribute of this technology is its safety profile. Okay, I mentioned that the safety was similar from part 1 to part 2. I should also mention, one of the things that we think is important about the GOG efficacy data from a 12-month overall survival is that it was consistent, right? So in science, we'd like to see consistent results. So, looking from part 1 of 38.5 and seeing essentially the same number in part 2, we think is another important finding. Next slide. So with these data in hand, as I said it a couple times already, we think it's important to take a run at a conditional license in Europe. We've laid the foundation to start to do that. We started doing that, as this slide says, in July. We applied for and received what's known as a CAT designation. This is the committee, I think, for advanced therapeutics. I guess the way I think about it is it's like a rolling submission opportunity in Europe. And so we've already filed the tox work and the CMC work with respect to what would be the equivalent of a BLA in the United States. So, as you see here, conditional marketing authorization may be granted based upon the risk-benefit profile in an unmet medical need. Bob Ashworth, who runs our regulatory group, is here tonight. He can provide a little bit more information of how many times that has been done, how many other therapeutic agents have kind of run that pathway. Bottom line is, based upon the data that we've already posted up or that's been seen by the GOG study, we think it makes a tremendous amount of sense to move for the conditional licensing in Europe. Okay, what about the United States? So I take a step back. The conditional licensing, like a conditional license in the United States, requires a confirmatory study. So you have to do a confirmatory study, agree to do a confirmatory study in order to achieve a conditional license in Europe. So our plan is, and actually announcing it here right now, is to do a metastatic nth line cervical cancer study that will be randomized. So think of the GOG study with a control. That's the study that we're going to start to do in the United States and globally. And we're anticipating doing that starting first half of next year. That's our goal. We have a phase 3 study that's already opening, and sites are being selected and opening right now. Our plan will be to kind of leverage those sites and make this study, phase 3 study, in the metastatic setting be a companion study to the study I'll describe in a moment, which is our aim to serve study. So that's essentially the pathway in the United States. We want to use that conditional study that I just described, which would be a phase 3 study in the metastatic setting, and get those data and look to see what registration opportunities that might afford us in the United States. We're looking to do that study as quickly as we possibly can. We don't intend to file for a special protocol assessment with respect to that study. We think it's a pretty straightforward study, and we think we have the opportunity to get that study done in a relatively efficient manner. So that is our goal. The other kind of parallel activities that are going on while we'll be doing that study is the study that Brian is the PI on. That's the combination with Durvalumab. So, folks have said to us, hey, as you're planning on doing a phase 3 study, wouldn't you just wait for that study to read out and then do a combination study as opposed to a monotherapy AXAL in that same setting? And our response is, one, it's going to take a little while for that study to read out. Brian will give some perspective on how long. Secondly, we don't think it makes sense to wait. If we wait for that study to read out, then we're going to be in the a while from now. But what made more sense to us was to say, let's start the controlled study now, and we potentially can add on to that study a combination arm if the data from the Durvalumab combination makes sense to do so. Okay, next slide. So this is the schema from our phase 3 study. We call this aim to serve. This study, from my point of view, is probably the sweet spot for immunotherapies. Why? These patients don't have disease. They are free of disease as they come onto this study. They have been treated with chemotherapy, radiation, plus or minus surgery, and there's no evidence of measurable disease as they get enrolled into this study. However, they are at risk for recurrence. So these patients are in a position where they have received primary care treatment, chemo, radiation, and they're being told by their physician, you're at risk for recurrence. The best we can do is watch and wait. We're going to do more MRIs and scans on periodic intervals and see what happens. The problem is, in that high-risk patient population, again, as Brian knows, is they recur. And when they recur, they die. The path there is a difficult one, and it entails a lot of platinum and various regimens. So as a company, we said, listening to Brian and his colleagues and people like Brad Monk, let's prevent them from recurring if we can, right? And in that setting, I really like the opportunity for a couple reasons. One, these are relatively intact immunologically patients. Two, we're not asking the technology as a monotherapy to knock down bulky established disease. And three, when you look at what's been done with the backbone technology in other settings, it seems that this technology has the ability to prevent micrometastases from gaining a foothold. And again, that's what we want to try to do in this study. 2-to-1 randomization, placebo versus AXAL. It's an event-driven study. Next slide, please. So in this setting, we did take a step back and say, let's measure twice and cut once. And let's go to the FDA and get a special protocol assessment. Our data was in the metastatic setting. Now we're asking the FDA to approve a progression-free, recurrence-free survival endpoint. And we felt it made a lot of sense to go to the FDA and have them review our protocol for adequacy. That's what we did. We achieved that in July, and it took months for us to get there. In fact, at the outset, the FDA basically told us, just so you know, this is not a short process. This process takes months. I personally was on the receiving end of a company that didn't take the time to actually get a special protocol assessment for their post-pivotal study, and it was a mistake. I think it's especially important for biotech companies like Advaxis to go to the extra mile and get that SPA assessment, which is what we did in July. After we had that, and actually slightly in advance of that, we were foot on the gas pedal to get sites open. We now have 3 sites open in the United States. We expect to have 12 by the end of the year. We're currently screening patients. As this slide says, we're expecting to open ex-US, that's worldwide, in the first quarter of 2017. There's people here tonight that are responsible for getting that done for the company. Chris Duke is our new Chief Operating Officer, is in the back. John Hayward reports to Chris, and he's also responsible for making sure that those sites get up and running. That's an important feature for us. Obviously, you have to have open sites to get patients on study. We've talked internally about how we want to provide updates with respect to this study. And we think the best thing for us to do is to stay consistent with what we've done historically, which is give updates at the beginning of the year and in the middle part of the year. Usually right around pre-ASCO and in the January timeframe. That's what we intend to do with this study. So here's the estimated trial timeline. We expect 50% of the patient enrollment to be achieved in the first half of 2018. As we provide those updates, as I said, in the beginning of the year and the middle part of the year, we'll provide some guidance with respect to how we're tracking against that target. Last patient enrollment is again estimated to be fourth quarter of 2018. Study completion first half of 2020. An event-driven study, 184 events. So obviously, those events are going to drive the study completion. Next slide. Okay, just in summary, the points I hope to have made tonight is, 1, cervical cancer is a major health problem. There is extreme little drug development in this setting. I think AXAL is exquisitely positioned to take advantage of that, both competitively, commercially, and for the benefit of patients and physicians who absolutely need treatment options in this setting. I hope one of my jobs over the next year and the coming months is to bring that point forward so people understand that this is a profound opportunity. Again, not only from an unmet medical need perspective, but also from a commercial perspective. Secondly, we're filing in Europe. We're looking to file that as soon as we possibly can. We said second half next year. We're trying to get it towards the middle part of next year. Again, I didn't actually mention the CAT designation saves time off of the review cycle for that. Generally about a year in Europe. So that review would probably be about 8 months or 10 months. Our goal is in 2018 to have a drug approval in hand, a conditional license in hand in Europe. Okay. Third point, sweet spot for monotherapy, immunotherapy is in those high-risk, locally advanced settings. And that's the purpose with that study, which is now underway. And I hope I've given you some timelines or some expectations around the timelines as to when we'll provide data updates on that. So I really want to thank you for listening to that presentation, but I'd also like to introduce our next speaker, is Dr. Brian Slomovitz. Brian, again, University of Miami, is one of the world's leading gynecological oncologists. I really appreciate, Brian, that you traveled up here to present here today. Brian is the PI on the study that's, again, going to be the subject matter of information provided tomorrow. Brian, thank you. Want the pointer? Thanks. You want that?
Okay, thank you. Dan, thanks for that introduction. And just a couple of points before I start my talk. The thing that's really exciting for me about this is that it's a drug. It's a combination. It's going after a disease which cervical cancer should be the first disease that's really being studied with this Listeria product, because it's such a good target. And I'll go through some of the preclinical data. And in the field of gynecologic oncology, oftentimes we're one of the later, and in women's health for that matter, oftentimes other diseases are studied, and then they see if there's a role to fit in for ovarian cancer or cervical cancer. And the fact that the company's really going after cervical indications, really, from my perspective, we as a community of gynecologic oncologists, we're grateful for that. G1 oncologists, just to let people know, they oftentimes don't realize it, there's only 1,200 of us in the country. So as a specialty, we're a very, very small specialty. We treat cervical cancer, endometrial cancer, ovarian cancer. There's only about 40 training sites in the country. And 1,200 of us. So I did my training at MD Anderson. I'm originally from New Jersey. And now I'm in Washington, D.C. on election week, so it's pretty exciting. I want to go through the data, and I want to go through this study. It's an exciting study that makes a lot of sense. In preclinical models, the combination of AXAL and the immune checkpoint inhibitor produced increased survival and significantly reduced tumor growth versus either agent alone. This study this preclinical work has provided the rationale for studying these agents in a combination therapy. Here's some just for rationale for investigating the two. AXAL is a live, irreversibly attenuated, non-pathogenic Listeria immunotherapy developed for the treatment of HPV-associated cancers. The checkpoint that we're using, Durva, is a selective high-affinity IgG1 monoclonal antibody that blocks programmed cell death, PD-L1, binding to the PD-L1 and CD80. Studies in mouse models has demonstrated that antibodies directed against PD-L1 or PD-1 do exhibit anti-tumor activity, and Dan shared with you some of the single-agent activity of these agents in the Keynote trials. So here's the rationale and the study design, really. The eligibility was HPV-positive metastatic head and neck cancer. So for the head and neck patients, they did require to be HPV-positive, and it allowed for all recurrent or metastatic cervical cancer patients. Measurable disease and evaluable disease was required, and they also required was a relatively good performance status. A lot of patients, we need to come up with a treatment for these patients. A lot of these patients, when they present, their performance status has excluded them for the trial. So it's that further emphasizes the unmet need and the need to come up with active agents for this group of patients. In Part A, all patients received the combination. We had 11 patients enrolled, and I'll go through the data. The AXAL was dose level 1, the 3 milligrams per kilogram. AXAL was fixed at 10 to the 9th dose, and dose level 2 was 10 mg per kilogram. 5 patients were in dose level 1, 6 patients in dose level 2. And then Part B of the study, which we've just started, goes to one arm is Durva monotherapy, and the other arm is a combination therapy. And we're looking to enroll 45 patients in each arm. The objectives are listed there. Here's some of the information on the patient characteristics of the 11 patients thus far enrolled, and this is what's going to be reported on our poster on Saturday. The median age was 52. Most patients had good performance status. 10 were 0. One of them was performance status 1. High proportion of white patients, mostly females, obviously, with cervical cancer indication, but with some male patients in the head and neck group. The prior lines of systemic therapy, you can see it's a relatively, it is a pretreated group for the patients that would be considered heavily pretreated. And prior biologic therapies, bevacizumab, it does have an indication in cervix cancer, so we see that a lot of patients did receive bevacizumab. One patient received cetuximab and one other therapy. Adverse events were generally as expected based on the profiles of each agent. There was only one AE that led to discontinuation, which was a grade 3 diarrhea. All deaths were due to disease progression. 2 patients had SAEs that were potentially treatment-related. Grade 3 adverse events were related to cytokine release syndrome and included rigors and/or chills, decreased white count, diarrhea, and shortness of breath. 1 grade 4 case of hypotension was managed and the infusion therapy was reported. The most frequent treatment-related AEs were chills and rigors, fever, nausea, hypotension, diarrhea, fatigue, tachycardia, and headaches, as listed in the table here. Grade 3 chills and rigors were reported in 2 patients, and grade 3 diarrhea in 1 patient. 1 patient had a grade 4 hypotension, as mentioned. The dose for the randomized phase 2 was declared at 10 mg per kilogram. This is based on the primary objective of the first part of the study and was to find the MTD. It didn't take into consideration any of the activity. So this is the results. Again, it's important to realize that this is an early phase trial, but these are important results to look at and to look at critically. These results are particularly encouraging. This is my patient right here. It's extremely encouraging. Again, this is a recurrent cervix cancer is a non-treatable disease. Most patients, when we talk to them, we don't have any treatment options. We try to do things that help them live a little bit longer, but to say that they're going to be disease-free is something that is unexpected. It's rare to see this level of response. We can see here, there's a head and neck patient that's stable disease on active combination therapy. What I want to highlight here is, in my patient, we all remember the clinical hold, and the patient received 1 dose, had a PR. Once she resumed AXAL, she went from the PR to a CR. So there was something about at that time point, something that happened on a biologic level that further enhanced the response, which was, from my perspective, it was very exciting. The clinical hold raised a lot of people's blood pressure, but to see this after it came off of hold was exciting. The patient who received a CR with the combination regimen had been heavily pretreated with aggressive chemotherapy. You can see she had 3 different lines of platinum-based therapy: cisplatin, paclitaxel, and bevacizumab, carboplatin, paclitaxel, and bevacizumab. She enrolled in the clinical trial and received 1 dose of the AXAL prior to the clinical hold. As mentioned, she then continued on the monotherapy. This converted to the CR when the AXAL was reinitiated. Therapy was and is well tolerated with her. She had transient grade 1 chills, fatigue, fever, nausea, vomiting, headache, and hypotension. She had 1 event of grade 2 rigors. One thing that's also fair to say, the study was initially designed to stop after a certain period of time. Because of the clinical hold and because the patient was doing so well and because of the patient's strong desire to continue, we were able to get IRB approval to continue her on therapy, and she continues to do well with the Advaxis and the Durva. Here's some images that I want to share, which are compelling. These are scans of the periaortic lymph nodes. Here's the initial scan, which you could see here. We have an enlarged periaortic lymph node. As we move 4 months later, it's beginning to shrink in size, calling it a partial remission. 2 months after that, it's continued to shrink, coming off of clinical hold, and then we have a CR, and the periaortic lymph node enlargement has gone away. Here's the same patient with our axillary lymph nodes. You can see here quite enlarged axillary lymph node, which is palpable on exam. Shrinking after 1 dose. Sustained PR, coming off of clinical hold, confirmatory of a CR. These data are highly encouraging. Again, the 1 CR and the activity that we're seeing is remarkable in this aggressive cancer setting, particularly in a patient who had heavily pretreated. As mentioned, enrollment in the expansion Part A and randomization to Part B has commenced for the HPV-positive head and neck and for all cervical cancer patients. Milestones of this trial: Part A, dose escalation is completed. In Part A, dose expansion up to 20 squamous cell head and neck cancer patients. Enrollment initiated. No new patients currently. Part B, randomized combination versus monotherapy, has initiated and currently enrolling. I think we have 3 patients that are on this currently at University of Miami. Completion of enrollment is anticipated in the second half of 2017, with initial preliminary data uptake in the Q4 of 2017. Thank you very much for your attention. I'll take any questions. Yes, sir.
I guess a
Yeah, no, that's a great question. And, the advantage from the Keynote study is that we sort of see what the single-agent checkpoint inhibitors are doing. And while they're doing something, it's clearly not enough. There's something about, when you take all the data together, when there's something about this preliminary data, extremely preliminary data, but with the one response, there's something, looking at that in combination with looking at the 265 data, which is single-agent activity. In select patients, there's definitely a signal, there's definitely activity of this listeria-based immunotherapy, which is, from a clinical perspective, is surprising, but we're seeing it, right? We're seeing patients that are doing better than they should be doing, which is quite remarkable. So I'm not sure. So there's something about both that are additive and perhaps synergistic.
I guess one way to approach it would be the NCI trial had this modeling of 25% based on the risk factors.
Correct.
That was for no treatment. What would it be? Is there a similar model for just the one component?
Right. So so a similar model of what it would be if it was equal populations of patients getting a checkpoint versus getting the listeria versus getting both sounds like a good trial design. The reality is these patients are, it's a relatively rare disease, so it's something that we'll need to look forward to. But in the absence of that prospective data, we have to take the data that we have available, and which demonstrates that there's definitely a signal, some activity, significant activity in the listeria-treated patients. Yes, sir.
Why are you using an unapproved PD-1 inhibitor when you have Keytruda and Opdivo with the AXAL?
My sense is that it wasn't approved at the time of the design, but I'll leave that to Dan.
Yeah, sure, thanks. We have two studies ongoing in combination with checkpoints. We have a PD-L1 study, that's this study, and we have a PD-1 study, that's with Keytruda in prostate cancer. So, as we looked at what therapeutic agent, i.e. a CPI, to combine here, we felt it made sense to take a look at both the checkpoint that was PD-L1, this candidate, as well as the one that was already established, which was Keytruda. The other, I think, kind of commercial point there is these are really, combination studies without inhibiting our ability to work with other therapeutic agents, i.e. other checkpoints. So this is a collaboration with AstraZeneca, but it does not require us to go further with Durvalumab. If, for example, we wanted to move into another, like I referenced before, looking to potentially add a checkpoint in the metastatic setting, we would not be precluded from looking to work with another CPI. Does that answer your question?
Yeah, it sounds like it's proprietary and AstraZeneca rather than, why don't you do a trial? Usually the FDA doesn't like using two unapproved products.
Yeah, I think that's so the question is, you've got to be able to show the therapeutic contribution of both of the agents, basically. I think would be the idea. And as you might recall the schema design from Brian's slide earlier, which showed that there's a Durvalumab only arm to the study and a combination arm. So the 265 study I showed you gives the contribution of AXAL as a monotherapy in this setting. The Durvalumab mono part of the study gives you an understanding of what the anti-PD-L1 does in this setting as a monotherapy. Great. Are there any other questions? Okay, Brian, thank you so much. I really appreciate it. Let's give that to Mike. So as I said at the beginning, Michael Princiat is the head of our research at Advaxis. Mike is a listeriologist, and I didn't actually know that word actually existed, but it does, and Mike is. So without further ado, Mike, if you can please take the podium. Thanks.
I'll throw a caveat in here. I'm more trained as an immunologist who's worked with listeria, and I guess by default that makes you a listeriologist, but I'm sure the real listeriologists out there would take issue with that. But, okay, I'm just going to take a few minutes up here to introduce you to our neoantigen platform that we have in development. Give you an overview of what that platform is, as well as touch briefly on what the landscape is out there with other companies that are also working on this. As I'm sure most everyone in here knows, there's a great deal of interest in development of targeting tumor cells using neoantigens that are present in the tumor and not present in normal tissue. And that's the premise for the development of this platform. So, okay, I didn't realize I had this here in my hands. So, okay. So, first slide, I'll just, for those of you who may not be familiar with the neoantigen platform, I'll just mention briefly exactly what this is. Tumors are known to develop mutations, and depending on the tumor type, the number of mutations they develop can be quite variable. These mutations in genes will, at least in some cases, code for different amino acid substitutions in individual proteins, which will show up in the cell cytosol, on the cell surface, or whatever the case may be. And these proteins at that particular spot will wind up appearing as foreign to the immune system. And what we're trying to do here with this approach is to co-opt the cellular immune system to recognize those mutated amino acids in the context of antigen presented on MHC class 1, so that the immune system can then target those mutations in the tumor cells. Okay? So immunotherapies work by activating patient's immune system, as it says there, and what you're really trying to do here is to activate high avidity cytotoxic T cells, and in this case specifically against neoantigens. Checkpoint inhibition works by enabling T cells that already exist in the immune system to be able to expand and become more active, if you will, more cytotoxic towards those neoantigens. But the idea here is that there are probably some, if not many, of the neoantigens that could serve as targets, may not be targeted in a patient who has not been immunized, because those neoantigens are just not expressed either in an environment or in a quantity that is high enough to be able to actually prime the immune system. So what we're doing with our platform is introducing these neoantigens into a very strong vector that primes the immune system. Once that immune system is primed against those neoantigens, those T cells can expand as cytotoxic effectors and then locate those neoantigens expressed on these tumor cells and target them for destruction. Okay? And because these neoantigens are expressed only on the tumor cells and not on normal cells, you avoid the problem of both central tolerance and also autoimmunity, if you will. Okay. So there is a great deal of activity right now in this space, if you will, in the neoantigen space. And also, I think, a great deal of excitement, and I'm hoping that Advaxis is contributing both to that interest and excitement. What I'm going to do here is touch briefly on some of the companies that are also working on neoantigens to sort of show you the different approaches that are being taken and where Advaxis falls into this landscape, if you will. Obviously, top of the list is Advaxis and our collaboration with Amgen, and I'll be talking in the next few slides about our approach. Gritstone has partnered with Immune Design. Gritstone is some of the physicians at Memorial Sloan Kettering working with Immune Design in Seattle are using a viral vector to target neoantigens. They use predictive algorithms to determine which neoantigens they are going to put into that vector. I will talk about predictive algorithms in a bit more detail momentarily. Neon, started by a number of the leaders in this field out of Harvard and a couple of other places. They are using, and they're not terribly detailed in their website as to their approach, but to the best of my knowledge, they are using peptides in combination with poly-IC as an adjuvant. Okay, they are targeting melanoma, lung cancer, and bladder cancers. some of the information, I won't run down this list about where they are in terms of clinical trials are listed on the right side here. BioNTech is a company in Germany that is taking a couple of different approaches. Probably their central feature is they are delivering neoantigens using mRNA, and they are using some predetermined, let's say, somatic mutations, antigens, in combination with ones that are pulled out of sequencing data for individual patients, and again, delivered by mRNA. They are doing this with triple-negative breast cancer. They are also working with a company called Advaxis, which is a company that really focuses on doing peptide elution from cancer cells and identifying neoantigens that way. And for those studies, they're using a peptide with poly-IC approach, similar to Neon's, coupling that with GM-CSF as an activator of the immune system. This is important, as I will discuss in a little bit more detail momentarily, is that most of these approaches require some type of adjuvant or adjuvants to prime the immune system and to activate the immune system to get an effective response using either peptides or mRNA approaches. Viral vectors are a little bit better at sort of kickstarting the immune system, if you will. There are quite a few trials going on at different academic institutions, and I'm not going to go through and list them. And for those of you that know the landscape, there are other companies that are coming forward looking at neoantigens using a number of different approaches that are in development and not quite as far along as the rest of these companies. And I will also mention, it's not up here at the moment, there is one other company, and it's the only other company that's using Listeria as an immunotherapy, called Aduro. They had a presentation yesterday where they introduced their work on neoantigens. They are a Listeria company like ours. There are many similarities between our platforms. There are some differences. The primary differences between what Aduro is doing and what we are doing with respect to neoantigens is that they are using a predictive algorithm, as most of these other companies are doing, to select what antigens they're going to put in from the data that they presented yesterday, and this is pretty much hot off the presses. They have a capacity of about 25 neoantigens with their vector system, and the one that they described yesterday, they included 9 neoantigens in that particular vector. Again, I will talk about this in just a little bit more detail in a moment. So, neoantigens. It's a personalized approach to tumor immunotherapy, okay? And basically what you are doing is you're letting the patient's unique cancer mutations tell the immune system how to target that tumor, okay? What would an ideal platform look like? Well, something that directly activates antigen-presenting cells through an attenuated but specific infection. That's, in this case, in our Listeria platform. Something that can deliver a large payload of neoantigens that are capable of producing a T-cell response, and at the same time, and again, this is an ideal vector, that can decrease the activity of regulatory T-cells and MDSCs in the tumor microenvironment. Ideally, it will be synergistic with checkpoint inhibitors, which can augment T-cell responses. And the manufacturing process, and this is important, must meet the requirements for scalability and especially rapid turnaround and cost. Because when you're talking about a personalized approach, you have to be able to sequence the tumor and the normal tissue and get those data back in time to design your therapeutic product and get it into the patient in a therapeutically meaningful time frame. And I will point out, and I'll talk about these data again in a moment, that Advaxis-NEO has been demonstrated to control tumors in an in vivo mouse model. I'll show those data at the very end. I'm not going to get into this in too much detail. I could sit here and, as Dan pointed out, as a listeriologist, immunologist, I could go on and on about this, but I'm going to point out the salient features of our platform, okay? Listeria, as you've already heard, is a gram-positive intracellular bacterium. The important features of this in terms of as an antigen delivery system is that once Listeria enters the host cell, and in the case of humans, the primary cell that infects is the dendritic cell, which is the main cell responsible for priming the immune response. Within a few minutes, it escapes the phagosome, enters the host cell cytosol, and begins secreting a variety of different proteins. Most of them are listerial proteins, but it also secretes, if you introduce it, which we do using a plasmid, it will introduce your protein of choice. In this case, we use a truncated form of LLO, which has a secretion signal at the amino terminus of the protein, and C-terminal to that, we put in our tumor-associated antigen. And in the case of ADXS-NEO, that is a collection of neoantigens on a string. And again, I'll show that to you in a little bit more detail. The TLLO TAA fusion protein is expressed from plasmids, as I just said, and we can have anywhere as upwards of 80 to 100 plasmid copies per bacteria. Each of these plasmids can carry a payload of 20 to 25 neoantigens. We're expressed as 21 amino acid, let's say, beads on a string for each one of the neoantigens. You can, and this is also important, you can deliver multiple constructs to one patient. So while you can put in 20 to 25 neoantigens in one construct, you can deliver up to 5 constructs per patient, each one containing a different set of neoantigens. That gives us a capacity in the ballpark of 100 to 125 tumor neoantigens per therapeutic dose, if you will. The other really important thing about the Listeria platform is because it is a bacterium, it really has quite an impact on priming the immune system. It expresses pathogen-associated molecular patterns. It activates STING, dangerous associated molecular patterns, NOD1, NOD2, TLRs, toll-like receptors, and so on. So it does a really great job of turning on both the innate and the adaptive immune system. The other thing about Listeria, also very important as a therapy, is that there is basically no neutralizing antibody response to the Listeria. It can be given repeatedly with basically no effect on its ability to continually boost the T cell response. And it's a characteristic associated with the life cycle or lifestyle of the Listeria bacterium that is almost unique to Listeria. And we have evidence that the TLLO antigen fusion protein decreases the activity of Tregs and MDSCs in the tumor microenvironment. So it is not just a vaccine vector, if you will. It is an immunotherapy that has other effects as well, and important effects from the standpoint of the therapeutic activity. Okay. As I mentioned before, we've just recently partnered with Amgen on this platform, on the NEO platform, and in the process of doing that, we've generated preclinical data as they were doing due diligence. We did a pretty good pressure testing of this system in a mouse model, and I'm just going to show you basically as sort of a proof of concept what we did. This is a tried and true B16F10 melanoma model that's often used in mice. This tumor expresses something like 3,000 or 4,000 mutations in the DNA, resulting in well over 1,000 non-synonymous mutations. A very extensive study done by John Castle and company went through and looked at these different neoantigens and pulled out about 50 using an algorithm, of which around 12 were shown to be immunogenic, and 2 of them controlled tumor growth. Okay, so we use this model to answer the questions of can we do it? Will this ADXS-NEO approach actually present neoantigens? And what will happen if you use that in a mouse model to control tumor growth? Okay. And in addition, we had other questions like, are more neoantigens better than fewer? And how does it compare with peptide and poly-IC vaccines? So what we did is we took some of the peptides that were identified by John Castle and company, and we we made one construct that included most of the immunogenic peptides, including what's called MUT30 and MUT44, and these are the two that control tumor growth. And we included the other ones that were shown to be immunogenic in this particular mouse model. But we also asked the question is, what happens if we take one of these peptides that can control tumor growth, but then we include, which was probably what's going to happen when we're actually making these vectors for use in humans, if we include a collection of other peptides that are not considered to be particularly immunogenic, will they diminish the ability to control the tumor growth if you're expressing these other peptides in the same vector as one that does control the tumor? And to cut to the chase here, this graph is from the AXAL study, showing what happens in a control peptide plus poly-IC, and then the MUT30 peptide that we just showed you that controls tumor growth with poly-IC. When we did the same thing using our vector, and we either, and these are all mice that obviously have the B16 tumor implanted, we either treated them with PBS or what's called LMDDA274, which is basically the listeria with that TLLO and no tumor antigen afterwards, okay? And then those two sets of peptides, one we called Neo12 with the immunogenic peptides, and Neo20, which is the MUT30 peptide, along with 19 others that really not associated with any kind of tumor control. And as you can see, the one with the immunogenic peptides did a very nice job, comparable or better than what was seen using a peptide. But what's really interesting here is when you put only one of the tumor control peptides in and introduce these others, you actually see what's bordering on statistical significance. You see as good or better control with what we call the Neo20 construct. So as a proof of concept, it works very, very well, and it's very encouraging to us in terms of moving into the clinic. And with that said, as I mentioned, we are doing this now in collaboration with Amgen, and this collaboration has been dubbed MINE, and I'm happy to take any questions.
Would you try to peptide in your model, or is it just comparing versus historical AXAL study?
So predominantly historical. We are doing some studies where we will put in peptide and peptide and poly-IC. But quite honestly, I mean, that's been done many times. There's a lot of historical data. We're really not trying to do a bake-off, if you will, with that approach. We're really looking to see what our vector does.
The neoantigen sequences that you mentioned were 21 protein. Sorry, is that the somatic mutations in the middle and you're choosing 10 to the left to the right? Like you see?
Right, correct.
And if so, then why don't you just take the 8 to 11 or epitope cleavage, like epitope location versus the entire 21?
Right. So, and I'm sorry, I basically alluded to that, but sort of skipped over, and that's a very, very good question. As I mentioned before, we are not using, one of the main differences, and it's actually one of the most important points here, is we are not using a predictive algorithm, okay? The reason for that, and I'm really glad you brought this up, the primary reason for that is that the predictive algorithms used essentially a system which has been trained on viral peptides, if you will, what will bind to MHC, class 1 molecules, HLA, mouse, whatever the case may be. And to say that they are somewhat less than robust in how they function, most of the data that's coming forth now is that those predictive algorithms in some cases will tell you what the immune response will be and what will control tumor, but in many cases when you use those algorithms, the ones that are actually associated with tumor control and or immune responses often fall way down on the list using those algorithms, okay? So we are using the capacity that we have with our vector system to include a large number of neoantigens so that we don't have to try and predict those. Because again, if you watch the neoantigen space, what you'll keep seeing is that a tumor had 80 neoantigens, and we used an algorithm to pick out the top 10, but the one that actually controlled tumor growth came out at number 40 or number 30 in that. We are getting around that and using an agnostic approach. For that reason, we do not predict what the 9, 10, or 11 mer is that will bind to that class 1 molecule. We put the non-synonymous mutation in the middle and put 10 amino acids from the wild type sequence, which is what will be in the tumor, both N and C terminal, and that basically covers any peptide that could be generated from that neoantigen that will wind up binding to a class 1 molecule. Okay? So it's an agnostic approach, and not using an algorithm, we will not define the exact sequence. When you do that too, you have to pick out the C-terminal binding residue, and we're trying to avoid that. Does that make sense? Okay. Yes?
Can we talk a little bit about the logistics involved in this type of treatment, this type of personalized medicine, in terms of the turnaround time?
Sure. Mm-hmm.
Facility, how long it's going to take for you guys to establish this?
Right. So the way it plays out right now is we get a tumor biopsy, we get normal sample, usually blood, okay? Send it to a provider that does the whole exome sequencing for us. The turnaround time on that is in the ballpark of a week and a half to maybe 2 weeks. They give us back the sequence data as well as selecting the non-synonymous mutations in a format that we've worked out with them that allows us to pick out the neoantigens and so on. So a week and a half, 2 weeks for that. That information is used to design the vector, which takes a day or two in-house. And that is sent off to a partner that we have that does the DNA synthesis. The DNA synthesis right now, depending on the sequence and the complexity and so on, can range anywhere from 2 to 3.5 weeks, perhaps. We are working with them to get that down to closer to the 2-week area consistently. They send us the DNA in the plasmid already made. That comes in-house within 24 to 48 hours. We transform that into the listeria, gets plated a couple more days, QC and so on and so forth, and it goes into manufacturing. And manufacturing is done in-house. We use a wave bag system for that. It's a closed system. It's bacterium. You inoculate and you have your product in 3 or 4 hours, literally that fast. It's a closed system, 10 mil bags. They're 10 mil bags, right? I'm on the basic research guy. I'm one of those geeky guys that, sits in the lab. So I'm speaking way outside, but I said enough of these things to be able to tell you the basics of this. And you get enough product basically for 2 years, okay, as well as all of the QC that's done with it for release. The entire turnaround time is in the 7 to 8 week ballpark, maybe hoping to get it closer to about 6 weeks. Am I speaking out of turn here?
Right. Miley, you great?
No, yeah, you're
Is that about right?
Yeah.
Okay. I don't want to get in trouble here. Okay. I'm speaking for other people, but that's the ballpark. And those are the major chunks of what has to happen. The time constraints are on the whole exome sequencing and on the DNA synthesis part of it. And everything's, from the point we get the plasmid, everything is done in-house. We do our own manufacturing. Because as a personalized product, being able to do that is really huge. And Mayo is setting up the manufacturing, has just done a brilliant job. And that's pretty much where we're at with that.
My question is about the potential utility of LM technology for cancers that have a low mutational burden, like pediatric malignancies or CLL.
Sure. And that's a really good question. As we mentioned before, and I didn't have that, obligatory slide from Schreiber and Schumacher but the mutational burden, I know, don't let anyone know I didn't put it in there, okay? I'll get in trouble. For lower mutation, everyone knows that checkpoint modulators seem to work better the higher the mutational burden. As I mentioned before, one of the reasons that may be the case is because tumors may not be very good at priming responses. And so you may need to have a lot of mutations to get 1 or 2 that can actually prime the system well enough to generate a T cell response. It is our hope that as we move forward, introducing these neoantigens into a vector system that is very good at priming the response will do a much better job of targeting lower mutational burden tumors. And that may not always be the case, okay? But we believe it will help because our vector system should do a much better job at priming lower avidity peptides, if you will, or peptide class 1 combinations perhaps ones that have lower avidity T cell responses that you just can't prime in the tumor microenvironment. But once you do, should be more than sufficient to actually then target the tumor. For those who aren't cognizant in how the immune system works, priming is not an easy thing to do, and it shouldn't be to avoid autoimmunity, but once you do turn a T cell, a cytotoxic T cell on to a particular peptide MHC combination, you only have to put 2 or 3 complexes on the surface of a cell to kill it. And that's the idea behind this. This is why introducing these neoantigens using a strong stimulator of the T cell immune response will hopefully get around that problem.
Good. Michael, thank you so much. Great. Just before we leave this slide, Amgen obviously diligenced the technology. Michael showed you a slide with respect to some of the data that was gathered as we went through that diligence process. I think it's important to know that Amgen had their pick of the litter with respect to these technologies and had diligenced many other technologies before they made the decision to partner with Advaxis on the technology. That was really rewarding in a way because they saw what Michael and Robert and many other people at the company have seen as the potential of this technology for years. So I think it's important, obviously validation, especially when you recognize Amgen as a company. They are an extremely powerful biotech company, and they've made the decision to partner with us on this technology. To that end, they're paying for the entire development program. So this program that we're now developing, moving into clinic, is the goal as soon as we possibly can. Amgen is paying for that entire development program. There are developmental milestones and sales milestones associated with the agreement as well as the back-end royalty. So we really appreciate the collaboration. We call it my Immunotherapy neoepitope, and we're particularly grateful for the fact that Amgen recognized what we recognized in this technology. Okay, so let's take a look at the road ahead. This is upcoming milestones broken down by the asset of the product candidate. AXAL in cervical, GOG-265, full data presentation first half of 2017. These are our goals. So we have, you've received the top line data from this study. You've seen the 25% 12-month OS, which is what the GOG calculated what they would have expected to see in an untreated patient population. And we're looking forward to presenting the full amount of data with respect to that study first half of 17. Filing GOG-265 with Europe in the European regulatory authorities second half 17. Aspirationally, we're looking to do that as soon as possible. I think realistically it's probably closer to the middle part of the year. Phase 3, high-risk locally advanced, aim to serve, enrollment update. ASCO, again, we've given you where we expect to be in the 50% target timeframe, and we'll give some context around the ASCO timeframe in terms of where we're at. AXAL-DURV combo, this is the study Brian presented. Updated data, second half of 17. So I think as I look at it, I think we'll have an opportunity at SITC next year to provide another set of data, obviously still preliminary data from the study, but basically more patients. Okay. Initiate phase 3 metastatic cervical cancer study. This is, as I said, we've announced for the first time this evening that we're starting a phase 3 study in the metastatic space. Monotherapy against physician's choice. 265, think of 265 with the control arm. Our goal is to get that up and running as soon as possible. We think we've got a little bit of an advantage in that we've already opened sites for aim to serve, US and Europe. Our goal is to really kind of make this be a companion study at those sites, and hopefully that will streamline getting sites up and running and get the study going. So I would say more to come in terms of study design, numbers of patients, and the like. Anal cancer, BRUG, this is Brown University is conducting a study in anal cancer. These are 11 patients that have already been enrolled. We've provided data with respect to those 11 patients. Historically, we're going to be looking to provide at ASCO 17 an update with respect to those 11 patients. A study that we call a Fawcett study, named after Farrah Fawcett. Farrah Fawcett actually created a foundation at the time of her death that supported the study that was done by Brown University. Farrah died of metastatic anal cancer. This study was named in her honor, and we are expecting to have stage 1 data in the first half of 2017 with respect to that study. This is, again, another 2-stage study. We'll be looking to open up the second stage of the study, second half of 2017. I had a next study that we've already provided data on at Baylor University, another Simon 2-stage study. It met the efficacy criteria, immunological efficacy criteria needed to open up into part 2. So this study is now opened up into part 2. It's happening both at Mount Sinai and Baylor. We provided information at AACR with respect to that study historically. We'll be looking to provide an update at ASCO 17. Per the question earlier, there's a combination study that's ongoing with Keytruda. Here are some of the upcoming milestones with respect to that study. Completion of enrollment of the AXAL, Advaxis PSA, Pembro initial cohort, end of 2016. Data from that initial cohort A and B, first half 17. Complete enrollment in the expansion cohort, first quarter 17. Preliminary data, first half 18. Again, estimates in terms of what we expect. Enrollment will drive those dates. HER2, this is a product candidate that's already been studied in canine osteosarcoma. It's now in a phase 1 study that's looking at basically HER2 overexpressing cancers. And we've got a preliminary data from the phase 1 targeted for ASCO 17. Importantly, the GOG has expressed interest to the company to do another Simon 2-stage study in pediatric osteosarcoma, predicated upon the data that was provided by Nikki Mason, who's a canine, is a vet who's already done work in this setting. And that work was the foundation for the GOG coming to the company and asking to do a study in metastatic osteosarc in a pediatric patient population. So our goal is to get that study started in the first half of 17. NEO is the program that Mike just went through and stepped through. This is, I think, a very important milestone for the company. Getting this IND filed and active is obviously going to give us our first opportunity to start treating patients against their own neoepitopes. Our goal is to have this done in the first half of 17. I think we're pretty far down that path, and I feel pretty good about being able to hit that. Something that we've not talked about before, ADXS hot. What is that? That's targeting hotspot mutations. So unlike NEO, which is targeting personalized, truly personalized approach, patient-specific neoantigens, this is going to be targeting relatively common hotspot mutations. So the difference will be, instead of building a plasmid derived from the information that's achieved when you compare the mutations in the tumor to a sample from the patient, a blood or tumor sample from the patient, and taking that specific information and building the plasmids around that, we'll be targeting what's known as hotspot mutations and having those plasmids built, and then have the patient inform us in terms of which plasmids and which of those hotspot mutations would be applicable to that specific patient. So unlike the NEO program, we're not targeting, exquisitely targeting a patient-specific neoantigens, but instead pre-building plasmids to hotspot mutations and having the patient inform us, which plasmid should be selected for that purpose. The goal there is to have the IND be ready in the first half of 17. Lastly, DT-LLO is an abbreviation for detoxified LLO. You recall from Michael's presentation, LLO is the half of the fusion protein that's secreted in the cytosol of the APC. It's highly immunogenic. It's a lysing protein. There's now data, preclinical data that's available and is going to be presented in December of 2016 that describes the use of detoxified LLO. When we saw that data, we made a decision that we wanted to start to pursue basically an ID aspect and component to the company. We asked Greg Mays, who's our Chief Operating Officer, or formerly our Chief Operating Officer, now our Chief Business Officer, to take charge of that program. And so that's something that Greg is currently working on. So again, a little bit of a long evening. I really appreciate everybody hanging in there with us. Brian is going to be here with his poster. Sorry, Brian, we got tagged you for a little poster discussion, if you don't mind. Obviously, many folks from the company are here. If you could all just raise your hand. Mayo is our manufacturing expert, so he's in the back and is available to answer questions. As I said, Bob is here. He's our regulatory expert. He's available as well. Thank you very much.
Excellent.
That's okay. Don't fall over.
No, excellent.
Good.
So tell what the metastatic phase 3.
Yeah.